but today I'd like to focus a bit more on two-photon optogenetics. And I appreciate that it's a very diverse audience, so I'm going to have short excursions on basic aspects of the topic throughout the talk. I'm going to start with a quick explanation of what optogenetics is, or what I mean with that. Hang on. And ultimately, optogenetics is a term that refers to the control of cell physiology by light using genetically encoded actuators. Those actuators are commonly retinal binding proteins, so-called opsins. And when these opsins have bound retinal, they're referred to as rhodopsins. And I say cell physiology and changing that. In neuroscience, ultimately, that almost always means membranes and membrane potential. And looking at two kind of hypothetical opsins here. One, we see transmembrane proteins. We have the extracellular space, which is positively charged, the intracellular space, which is relatively negatively charged. Membrane biophysics would dictate that those charges tend to accumulate close to the membrane. We have two transmembrane proteins, a channel and something more like a pump. If we shine light on those, using blue light for the one on the left, the channel, and then more amber light for the pump, what happens is they become active. In this case, it's simply, they simply compensate each other. In practice, obviously, if you have the channel on the left-hand side, so the classic channel rhodopsin, you'd see positive current streaming into the cell, or positive charge coming into the cell depolarizes and will at some point fire an action potential, and vice versa. Over here, we have negative charge being brought out, which again would depolarize. Very commonly, though, what these pumps will do, in the case of, for example, halorhodopsin, is they bring protons and other positive charge out of the cell, further hyperpolarizing it and inhibiting it. And indeed, if we look at some data from really the early days of optogenetics, cell here expressing halorhodopsin, which is activated by the yellowish light, and channel rhodopsin 2 activated by the blue light. When they patch onto the cell, they do a voltage clamp recording, they shine yellow light onto the cell, they see an outward current. So if the potential was free drift, the cell will become more hyperpolarized. And when they shine a blue light, they see an inward current, the cell becomes more activated. If the membrane potential is free to change here, what this would be a depolarization. And indeed, if they just let the cells freely change membrane potential, go ahead doing just voltage recordings, what they see is that with flashes of blue light, they can induce individual action potentials or spikes. And in parallel to that, when they shine the amber light as well, they deactivate the cell, they inhibit it, and they can block those spikes. Now, that's a nice simple way of activating and inactivating cells and controlling what they do. But if we want to go in vivo, we have to get these transgenes expressed. Now, there's obviously the approach of using transgenic animals, but that tends to be quite an investment. You need to do quite a bit of breeding. You have to maintain those colonies. That's quite a steep hurdle to climb. You can also use wild-type animals and just inject a virus that encodes for the opsin. And in many cases, what you'll also have, it'll co-express the opsin and a reporter gene that stains the entire cell, because the opsin tends to be expressed really only on the membrane, because it's a membrane-bound protein. So these viruses transfect, the constructs are in the cells express, and the cells become green if it's a green marker, red if it's a red marker, and there's a non-fluorescent or hardly fluorescent opsin in the cell membrane. And you can go ahead and repeat the experiment I just showed earlier, and you can get more specific. If you don't want to get all the cells but subcells, what you can do, a fairly efficient approach, is to use a Cree reporter line, which gets you the means of addressing targeting specific cells. Now you have, again, a virus, but this time the transgenes are sort of floxed in there. They're under Cree control. If the virus is injected, the cells get transfected, but only in the cells that are expressing Cree recombinase are they actually transcribed, and then you get protein expression. So that's all nice, and we've got everything working in terms of expression, but how do we now get into deeper tissue. I said, right, we want to do this in vivo. Ideally, we want to do this in a live mouse in vivo, possibly even an awake behaving mouse. And so we need to get deeper into the tissue. We don't want to just hit tons and tons of cells by shining a lot of blue light on there. And this is where multi-photon, or specifically two-photon excitation comes in. And again, a small excursion. I apologize to those who know this already, but I thought it helps kind of set the stage for the rest. And if we look at excitation of fluorescence, and there's a lot of photostimulation of opsins is similar or follows analogously here. We have the electronic system. We see a ground state, an excited state, and the molecule absorbs a blue photon, is excited. There's a bit of vibrational relaxation, and at some point it then comes to ground state and emits a photon. This is a wonderful quantum typical process. There's a single quantum jump. You need to absorb a single photon of blue light, and if you use light of different wavelengths, it just won't work. You won't get this defined energy gap bridged unless, and this is something that Albert Einstein predicted very early on, and then Maria Gerbert-Meyer went ahead and worked out in substantially more detail, put down the proper framework and formalism for this. Unless you have these two photons with half the energy, so double the wavelength, so instead of blue light, you're looking at far red light now, that interact with the molecule at exactly the same time. And again, you get your excitation into the excited state, or promotion to the excited state, and then it hangs around. And if it's an opsin, it just conducts current or acts as a pump until it returns to the ground state. If it's a fluorophore, it emits fluorescence. And how it got to that excited state doesn't matter once it is in that excited state. And this kind of already hints at two key advantages of using two-photon imaging. The first is the wavelength, and the second is the fact that you need this interaction with the molecule at the same time. So you need a very strong concentration, so to say, a very high density of excitation photons. And the first is simple. It's a little photospectroscopic experiment, so to say, that most of us did as kids. You take a flashlight, a torch, hold it behind your hand or in your mouth, and you see that you put white light in, orange, red light gets through. If we had an infrared camera here, we would see that near infrared light gets through even more efficiently. So that means that by using those longer wavelengths, we're able to penetrate deeper into the tissue. We can get our excitation spot deeper in. The other component is much more interesting and much more powerful. Is if you look at what we have here is a beaker of fluorescein solution. There's an objective you can just barely see hanging in there. There's a blue laser light, 48 laser line coming through, and you see this typical double cone of fluorescence excitation. Everywhere where there are blue photons, you get absorption and green fluorescence. Now with two-photon excitation, I said, you need that higher density. You'd expect that only really in the focus do you have enough excitation light to get photoactivation, to get photostimulation. And indeed, if I take the setup and all I do is change the wavelength of light coming through and mildly reposition the camera, but it's exactly the same system, you see that you only get fluorescence in the middle spot. So now you have this so-called optical sectioning. There's no out of focus component. You can now go ahead and target individual points in three-dimensional space in deeper tissue. And indeed, that works with optogenetics just as well as fluorescence. What we have here, this is work from David Hank's published just over 10 years ago. This is a cell in culture expressing YFP in the cytosol and channel rhodopsin 2 on the cell membrane. They scan over the cell and record both the current of the cell as well as the fluorescence. And you see whenever the laser line crosses the cytosol, you get nice yellow fluorescence, and whenever it crosses the membrane, you get an influx of charge into the cell. And you can tell that that charge keeps coming, even though the conductance lasts longer than the laser is on the actual membrane. That's because these opsins are quite slow. Once you switch it on, they stay on for milliseconds very often. But you can also tell that this is not a lot of charge. You're not going to really get a cell to spike with 10 picoamps of current coming into a cell. So what they did is they tried different ways of rastering over the cell. In the first instance, they very quickly rastered over the cell in a very fine raster pattern. It's a very slow, not very high excitatory postsynaptic current that they see. They go ahead and they do this faster, but with a with fewer lines, and they start speeding it up, and then they're really just zigzagging coarsely over the cell, and it's getting better, but it still doesn't look very much like an EPSC in a cell should, not the kind of current you'd expect from an actual synaptic input. And then they go ahead and they spiral over the cell instead. You can imagine that spiral pattern is basically tracing along the cell membrane, and now they're really recruiting a lot of current, and this will actually go ahead and stimulate cells. So in contrast to a one-photon case where you just shine blue light and the whole cell just spikes almost instantaneously, you need to build up that charge, and you need to build up that current by spiraling over the cell. You see, you get these actual potentials reliably, but there's a bit of a delay on that. And this basically means you can hop from cell to cell, spiral on a cell, hop to the next spiral on the cell. And this is something that most laser scanning microscopes will do. Our hyperscope has one beam path that you can use for imaging, and the other hidden behind here, identical to this one, can be used to actually spiral that excitation light over a cell. But if I think about working in vivo and then working in live animals, that's not the way the brain works. It's not a cell here, a cell there, a cell there. It's dozens to hundreds of cells at the same time becoming active. So what you really want is you want to hit multiple cells at the same time and spiral over them. So you want dozens to hundreds of laser beams going in. And you can't go ahead and just take dozens or hundreds of lasers and fire them. You need means of splitting this beam into points that focus into different positions in 3 dimensions, so that you can go ahead, activate those cells, get activity in those cells, and move on. And this is where so-called spatial light modulators come in. Now, when we talk about a spatial light modulator, that's what people from a more technical background will often call an LCOS, a liquid crystal on silicon spatial light modulator. You have a silicon chip, you have pixels of liquid crystal on there. What they do is they reflect the light, and apologies if I'm oversimplifying, but I'm hoping to give you a kind of rough idea of what's happening here. They reflect that light, and they have the ability of reflecting it kind of at different points with a different delay. So you're shifting the phase of the light. So if your incoming light, this is laser light, it's a nice flat wavefront, it's all coherent and is reflected, and you have a kind of phase mask on here, a pattern on this SLM, what you get is a distorted wavefront coming out, which is nice, but doesn't really help you much until you add a lens and you focus this. Then you get ultimately constructive and destructive interference, or not formally a Fourier transform of this wavefront, and you're targeting different spots, in this case 2 dimensions, but you can imagine if this is actually not a flat beam, but a proper laser beam, that this will be in 3 dimensions. And you can do that to target numerous spots. And now you're achieving exactly what I said would be needed before. And this is done in a module that Scientifica launched at the beginning of this year, the HoloStim 3D. HoloStim because this technology is called holography, and because it works in 3D. It's in addition to the hyperscope, bolts on, and then basically generates these patterns and that relays them over a set of galvanometric mirrors, so that you can then add the spiraling with those mirrors and spiral over all the neurons at the same time. And in practice, this works very well. What we have here is a view into the mouse brain. The cells here are expressing GCAM6, I believe, GCAM6M, and a redshifted version of channel-rhodopsin, C1V1. And when we then go ahead and monitor the activity in these cells while activating the light with the HoloStim, you can see these cells live. We can do this with these cells here, and you see there's quite a lot of activity, there's quite a lot of calcium activity being generated, but we can also do this in, sorry, this is just a repeat of what I had. Next slide, here we go. We can also do this easily in 100 cells. And here you see that there are a few cells that are quite bright already, but the majority of the calcium indicator expressing cells are dim until they become active. And this is 100 cells, and you can see that you get reliably on every stimulation, you get most of these cells responding. Now this is in vivo, so the cells do have background activity. Some might be a bit more inhibited, some might be a bit more excited, so there is variability between them, but in principle, this works really nicely. And where this really opens the door for the next set of experiments is that we're stimulating these cells, we're imaging this activity. And this is something that's not science fiction. It's being done by increasing numbers of neuroscience labs worldwide. And our main aim really is to make this technology easy to use, to bring it to more labs. But you can then go ahead and rather than just using activity to stimulate this as the stimulation to activate the cells, you can read out the activity using calcium imaging and use that to close the loop. And you can do actual closed loop photostimulation of these cells and use that to do what kind of the phrase that's being coined is all optical interrogation of neural circuits. And yeah, I'd finish here a short primer, which is kind of what was suggested we provide. And I'm happy to answer any questions that anybody might have. Thank you.
Okay, thank you very much, Christian. Very interesting primer, as you put it. So I have a question. I'm sure people will have questions in the thing, but I have a question about, you're talking here about, you mentioned you have a spot and you can scan it around in a spiral. There are then the holographic aspect of this. There are also other aspects of light shaping people have used for optogenetics, including, say, spatiotemporal focusing.
Yes.
And so on. What do you think of there's a variety of these methods here. Do you think they're all complementary and usable in different applications, or do you think that they have very different uses? Do you think we need all of them or just some of them?
I think they all have a good reason for existing. I think there's value to all of them. I think they have their trade-offs. So if we're looking at two-photon optogenetics, it's basically there's this typical debate is spots versus blobs or spirals versus disks. What I presented here it falls under the spots/spirals. What you can also do, what you hinted at, is you can do proper holography, generate cell-shaped disks, and activate only there. Now, the issue there is that two-photon excitation is very sensitive to the density. Obviously, if I have a certain amount of laser light I can put into a specimen, a biological specimen, before there's just too much heat. And that's kind of the starting point. We then have to figure out how many pixels, and again, I'm apologize to people who look at this in a more formal manner, but if for me, simplistically, biologically, from a biologist's point of view, I'm splitting the light into pixels. And I can split into a certain number of pixels before I just don't have the photon density. And I have the choice of splitting this power into 100 individual points, say, or 5 shapes to 20 pixels each. And so the advantage with this approach is I'm able to target larger number of cells at exactly the same time. But I need to spiral over those cells for a longer amount of time to actually get my activation. If I go for the disk or blob approach, I spread out the light, I hit whole cells, I basically paint the entire cell with light, I get excitation there, I get very quick buildup of current or charge, and I fire them cells very quickly, but I'm not able to access as many cells simultaneously. Now, in the past, the rule of thumb kind of was that at the end of the day, you're going to get roughly the same number of cells over 5 seconds. And it really is just how you want to spread that in time. There have been some changes. A lot of these options have gotten very fast, so that even with the spiraling approach, you actually can drive them to spike in about 2 milliseconds. So you can get quite quick. So Crimson is one of the ones used there. And I think then the trade-off becomes a bit more complicated. If you're interested, but I think it really remains to what it is. If you're interested in activating a large number of cells simultaneously, this approach that I presented here is the way to go. If you want to get as reliable as possible and as quickly as possible, a small number of cells to fire, spot disks are the way to go. There is another complexity here, and that's just instrumentation-wise. 3 out of 4 manufacturers or companies that have solutions like this on the market went for this approach. We're one of those 3. The spatial temporal focusing approach is one that only one company does, and the reason ultimately is that by the time that you're generating multiple arbitrary spots in arbitrary positions in 3D and add that temporal focusing to ensure that you're really getting a nice tight optical sectioning, you're using a method known as 3D shot as kind of the leading one right now. And at that point, you've got 2 SLMs plus a grading in sequence. It's not a cheap piece of kit, and it is it's a finicky piece of kit to maintain. So I think the reason that most companies like us went for this approach, use a single SLM, hit a bunch of spots, is because it's ultimately an affordable way of achieving the goal. Sorry, very, very long reply to...
No, very packed with information, so it's very useful. Thank you. So we haven't seen if we have any more questions in the chat. I don't see any popping up there. Yeah, so I mean, you may have already partially answered this question, I think, in that last one, but my other question was, certainly from my perspective, I'm very familiar with this from research labs, but you know, what stage are we at in terms of commercial availability of these things, and how easy is it? So how easy is it to get this into the hands of people who want to use this technology but really aren't instrument developers?
So the first of these systems, where an SLM sits on for this kind of stimulation, as I presented it here, on a commercial microscope readily available, I think that was launched in 2016 or so. We've since seen there was another company that was Brucker, then Thor Labs launched one, and now we recently joined that group. And we have the small advantage of being the late to the party, in the sense of by the time that we got to doing this, we already have the current versions of SLM, so we have slightly higher, well, not slightly, we have more than double the resolution. So the phase mass that we can generate are tend to be more complicated, but ultimately there are right now 3 of these commercial solutions available. As I said, there's one for the temporal focusing approach that's available, but I believe the main focus there really is one photon stimulation. The 2-photon side is the actual adding of the temporal focusing seems to be more an additional component that they do. If you go to the extent, the high-end, I'd say, solution, the really complicated one of a 3D shot, it's a Tinker solution. But if your aim is to stimulate dozens to hundreds of neurons simultaneously, there are a number of solutions out there, and we hope to convince people that the one that we have is one of the more usable ones.
Yeah. Okay. And just one final question from me, in case, see if anyone else comes up with any questions in the meantime. How critical is the speed of the SLM here for the applications? Because I think many people who work with these realize that there are a large number of relatively standard SLMs which are at the lower end of the price scale, and yet there are more specialist things which go way up the price scale. And if you were sort of implementing these things, it would be much nicer if you could deal with one of these which works, say, the 50 hertz kind of level.
Yeah.
The standard operation now.
All 3 manufacturers that are doing these have gone for ultimately quite fast SLMs. The point is, as you'll appreciate, if I compare an SLM to something like a DMD or a deformable mirror, an SLM is actually still slow, even the fast ones. And you want to be in a position that biology is your limiting factor. So I want the time-consuming factor, the bottleneck one, is ideally the time it takes the cells to actually spike and do whatever they do. So I want to be able to switch fast enough. So ideally, you want to be in a position that you're looking at a refresh rate of upwards of 100. I think most of them are now in the range of 200 to 400 hertz, because that way you know that the majority of the time is actually spent stimulating cells. Yeah.
Okay, great. Well, thank you very much, Christian, for your talk and your very informative answers to the questions there. Very useful.